Good afternoon, everyone, and thank you for the very kind introduction. I'm very happy to be here. Thank you for inviting me. I changed my title to On Precision Dieting and the Microbiome, given the topic we're discussing today. And as you've heard, my two groups and I have been studying host microbiome interactions for quite a long time. And I think it's fair to say that in the last 2 decades of the field's existence, many associations and correlations have been made between microbiome changes and almost any multifactorial human disease that you can think of. But what we try to do in our groups is to try and go beyond correlation and prove causality and mechanism, and we do this through development and utilization of a number of experimental and computational modalities that help us to characterize the microbiome at substantial resolution and to also prove causality. And in doing so, I think that our slowly maturing field has came to the realization that one can regard the microbiome, and I'm mainly focusing today on the gut microbiome, as a signaling hub that integrates into it many signals that come from the human host. For example, our immune system, our genome impact how our microbes are shaped and behave, but also almost any environmental factor that you can think of transmits signals to the microbiome. The microbiome integrates these environmental and endogenous signals and communicates them to the host, and we see our mission in life in trying to decode the alphabet of this language by which we and our microbes communicate in health and in disease. And of the many different factors that impact host microbiome interactions, diet is the most dominant one, the number 1, 2, and 3. And so for 2 decades, this has been one of our main foci of interest. And in the next few minutes, I'll share with you a couple of vignettes from our own work and discovery that I think would be very relevant to the cancer field and relate to how we view dietary microbiome interactions and their potential impact on human health. So I'll start by talking about the obvious, how dietary composition, how the composition of what we eat may impact the microbiome, signal to the microbiome, and impact our health. And when I started getting interested in this quite naive question over a decade and a half ago, I took the liberty of reading a lot of the nutritional literature that forms the basis on everything that you and I do in our daily lives when we try to come up with what we hope would be a healthy diet. And what was striking to me is that everything that we do is based in one way or another on grading systems or scoring systems that give different numbers to different foods. So for example, calories is one such very popular scoring system. We give numbers, we try to mix and match them and come up with what we hope would be good for us. A glycemic index is another scoring system, grading foods based on their ability to spike sugar levels in different individuals. So if you look in your smartphones, you'll find endless tables giving glycemic indexes to any food on Earth. But here comes the very big problem. When we repeated these experiments, we found that these food-related scoring systems are inherently flawed. Our eureka moment came over a decade ago when we naively studied very commonly used nutritional supplements, non-artificial artificial sweeteners or non-nutritive sweeteners that are consumed by hundreds of millions of people worldwide in trying to sweeten our foods without paying the caloric content. We wanted to see whether these seemingly inert food supplements actually impact the human body through their effects on the microbiome. And to make a very long story short, one of the experiments or some of the experiments we performed involved giving identical amounts of any given sweetener, for example, saccharin, to groups of humans. And what we were surprised to find that half the humans spiked their blood sugar levels in response to these sweeteners, and half didn't care at all, which was a eureka moment because it meant that giving a number to saccharin does not mean anything in the personalized manner if one person reacts opposite to another person. And to expand this beyond this early example, when we did this with all foods, we've now profiled over 100,000 people using the method I'm about to share with you. We found that people's response to any given food has a huge variance between individuals. So what that means is that rather than giving foods different numbers, we need to find a way of scoring the people that eat the food, which gave rise to a very ambitious study, which we termed the Personalized Nutrition Project. It involved at the beginning 1,000 people. We collected from them everything we could back a decade ago. including all the things you can see here, in continuous glucose monitor that monitored their blood sugar levels every 5 minutes for an entire week, and of course the microbiome, because this is what we do. And then a very smart group of people at the lab took all the data from each of these participants and used deep learning methods in order to develop algorithms that would predict a person's blood sugar response to any given food, even foods that they haven't consumed during a week of follow-up. And to summarize this 5 or 6 years of work into one slide, we were able to demonstrate that this data-driven personalized approach dramatically outperformed the one-size-fits-all dietary recommendations that we all follow in our daily lives. And this was validated by validation cohorts that were not part of the training, as we do with machine learning and deep learning. And we were quite happy and surprised by these results, but it was also time to put ourselves to the real-life test. So we engaged in a number of short-term interventions in which we took pre-diabetic individuals, we randomized them to receive these counterintuitive, personalized, good or bad diet that were so counterintuitive that a good diet for a person could include some chocolate and a bad diet could include sushi. Until you did these measurements and collected the data, you couldn't guess. And then we asked them to eat their individualized good foods for a week, their individualized bad foods for another week while we measured them. You can see here an example of a continuous glucose monitor of one of the participants. And you can see that during the week of bad personalized diet in red, that person spikes their blood sugar level, while switching them to a personalized good diet that could include some chocolate, the green graph was completely flattened. And when we revisited these deep learning algorithm in retrospect, they never asked for my permission on which feature to use for their prediction. We were pleasantly surprised to find that there were big chunks of host data, but also features coming from the microbiome. In fact, today, we can only measure the microbiome and a limited set of human features in order to get to a similar predictability. But the burden of proof was still on us, and we wanted to do a long-term study. We performed a randomized control trial on 200 pre-diabetic individuals, randomized to either get the best of the best Mediterranean diet, the gold standard, or this algorithm diet. And we followed them for a whole year. You can see here 3 different readouts of glucose control in these individuals. And you can see in red that the one-size-fits-all Mediterranean diet does have a good effect. It does improve glycemic control, as has been reported for decades. But I think you can already appreciate that this data-driven, personalized approach in green outperforms the one-size-fits-all diet, at least for this readout, in a quite substantial manner. Just to complete the story, I've started with our work on artificial sweeteners. It followed up with extensive testing by us and by many, many, many others of all the sweeteners that you guys buy in your local supermarkets. And this culminated a couple of years ago in the World Health Association issuing its first ever warning, global warning against the indiscriminate use of these substances, especially among populations at risk, such as diabetics, and so on and so forth. And where do we go from here? There have been published a number of validation studies, mainly from the developed world, validating the concept and adding knowledge into it. We are following up with this personalized or precision nutritional effort and carefully diving into non-metabolic disease, including inflammatory disease and lately cancer. And your NIH have initiated a flagship project, lots of money, a couple of years ago, and we hope that it is maintained and that we will learn more about, I think, this interesting new concept. So this was an example of how the composition of diet impacts our microbiome and our health. Equally interesting was another accidental finding by our lab, which is that dietary timing has very unique impacts on the microbiome that have their own downstream effects on the host. And this was another naive project in which we wanted to answer the question on whether the gut microbiome features a circadian activity. And to those of you interested in that exciting field, we know that every cell and every organ in our body features a circadian clock. The Nobel Prize was awarded for the discovery of the clock in 2017. And since we regard the gut microbiome as a neglected organ, we asked whether the gut microbes also follow this 24-hour cycle behavior. So this resulted in a very painful project for my postdocs. They had to collect poop from mice every 4 hours for consecutive days. And then we sequenced it, and to our surprise, we found that the commensals actually feature a very robust circadian activity or diurnal activity. You can see here one of the commensals going up during the night, down during the day, up during the night, down. You can measure them forever. They will continue to behave this way. You can see here a few dozen microbial functions. They go up during the night, down during the day, and so on and so forth. Now, this was very peculiar to us because if you think about it, these microbes, they live in the dark, so how the hell do they know that it's day and night and change their behavior? This resulted in 3 years of work, and at the end we found that the dietary timing is the major driver of this behavior. So when we're awake and we eat, the gut microbes feel that we're eating and change their behavior. When we're asleep and we don't eat, they change their behavior accordingly. Just to give you one example of one of many experiments in which we proved this, we took groups of mice, we force-fed them only during the night, and in another group, we took the mice and force-fed them only during the day. We sequenced the microbiome consecutively, and you can see that we could switch the circadian or the diurnal activity by 12 hours just by switching the dietary timing. Why is this important? It's important because for many, many thousands of years, humans and all mammals have been hardwired to follow a very strict circadian activity. We wake up when the sun rises, we went to work or whatever, then the sun set, we all went to sleep. But then during the Industrial Revolution, in a glimpse of evolution, all of a sudden we beat the circadian clock. We have light bulbs and electricity, so all of a sudden humans work in shifts. We have airplanes, we can fly across time zones. And with these disruptions in circadian activity came a huge risk of these chronically disrupted populations to develop obesity, type 2 diabetes, and even cancer. But we didn't know why circadian disruption is associated with these risks. So we wanted to ask whether maybe this interesting microbiome circadian activity could contribute to this impact. So we performed a large group of studies in mice in which we disrupted their circadian activity by switching them from light to dark cycles, generating one big jet lag in these mice. We also performed a very cute set of experiments in humans. I took a group of my postdocs and I flew them all over the world, economy, and all I asked them is to collect their poop before, during, and after they were jet lagged. Okay, so we sequenced their stuff, and of course it was greatly disrupted, but we really proud ourselves on trying to reach causality. And to reach causality, we transferred the jet-lagged mouse or human microbiomes into germ-free mice. They don't have any microbiome of their own. And to our surprise, they developed a quite dramatic tendency to develop obesity and type 2 diabetes, suggesting that the microbiome may participate in this risk behavior. And we and many others are engaged in very ambitious human trials now, trying to further elucidate and harness this knowledge towards human prevention and treatment. And the last piece of data I want to share with you relates to what I feel is the big elephant in the room, which is with all of these experiments in mice and in humans, what we learned is that we have a very hard time in accurately assessing the dietary exposome, especially in humans. Now, I have nothing against food frequency questionnaires, and food frequency questionnaires have transformed dietary and nutritional sciences in the last 3 decades by pioneering work by Dr. Willett and others, and they enable an affordable estimation of dietary intake, but they're also associated with many, many big challenges. People forget, people lie, people are ashamed to report on what they actually eat. When you look at these questionnaires, they're very laborious and people become tired. When we do multinational cohorts, the validated questionnaires differ between countries. And I can continue on and on. There are problems in getting to a single nutrient resolution. So if we could complement these food frequency questionnaires by an objective and non-invasive way to measure dietary impact or intake, I think it would be a great benefit to the field. Now, you all carry your smartphone, and you may ask me, why not take pictures of the foods and use AI and blah, blah, blah. But, and we've recently reviewed this. It's very promising, and it will come into use in the coming years. But my argument is that today, these modalities are mainly able to delineate foods that are very discrete, but they have no way of, telling us what's in our broccoli pie. So the short story I'm going to share with you relates to a completely different question. Again, an accidental finding. I had 4 champion postdocs in the lab that were trying for many years, 7 years, to find a way to directly measure the functional microbiome. Now, all of us in the microbiome field have been using DNA as the way to characterize these large and unculturable populations of microbes, but we kind of forgot that the DNA level only provides the potential for function while we disregard transcription and translation and all of that. So we wanted to see whether we can do this with the bacterial protein level, which is a huge task because the reference databases for proteins are giant, and there's lots of overlaps between proteins coming from different bacteria. So to make a long story short, the way we solved this was by a method called metagenome-informed metaproteomics, which consists of taking a sample, let's say a fecal sample, sequencing its microbial DNA by shotgun metagenomic sequencing, and using this as the per-sample reference database for the mass spec-based metaproteomics that is generated from the same sample. So it's quite expensive, but enables to dramatically increase your resolution because your reference database is much more constricted, and to shorten your running time and your expenses. And for the same price, we get from the mass spec the host metaproteomic response to different microbiomes. So for us, this method enabled for the first time to look directly at the host and microbiome function and their interactions. And using this, we were able to get a functional assessment of these interactions, to discover new biomarkers, therapeutic targets, and this deserves a different talk. But the bottom line is that we were able to annotate 97% of all proteins in a given sample to their origin, to their species origin of the microbiome. So I was very happy. I said, okay, let's publish this. But my postdocs are much smarter than me, and they were bothered by the fact that between 3 to 5% of all proteins in a given sample could not be annotated not to the microbiome reference database and not to the human reference database. And their idea was that maybe these proteins actually come from diet, which for us was an exciting opportunity maybe to generate a first-of-its-kind non-invasive way to measure the dietary exposome, the dietary intake in feces. So we've generated a third reference database consisting of unique proteins that come from hundreds of foods that are consumed in a developed country, and we projected our metaproteomic data on it, and indeed all of these missing proteins came from diet. Now, the burden of proof on proving that this could work was on us, and we started at the population level. So I took advantage of the fact that I have 2 labs, one in Israel and one in Germany. We allocated cohorts of healthy individuals in both countries. We performed our analysis on their fecal samples, and we looked at the dietary signals. So, for example, when we looked at the wheat signal in their poop, Israelis eat pitas, Germans eat pretzels, but wheat is wheat. So the signal was comparable between the 2 countries. But then we looked, for example, on pork, and we saw a huge German signal and 0 Israeli signal. And this, of course, stems from the fact that all of us in Israel are either Muslim or Jews, so we just don't do pork. So this gave us the first hint that this may be working. And what was even more exciting for us was that this unique pork signal didn't just come from one unique pork protein. In fact, in this particular case, there were 14 different unique pork proteins that all behave along the same direction, making the signal very trustworthy. Just to complete this story, people in Israel don't suffer of malnutrition, unfortunately, so they compensate for the lack of consumption of pork by eating much more chicken, and this could also be captured by this method. We then went to the individualized level, so we performed many experiments. For the sake of time, I'll just show you a couple. We took a group of healthy volunteers, asked them not to eat bananas, apples, cucumbers, and peanuts for the duration of the study, and then we gave them pairs of these foods under predefined days, and we performed this analysis on their fecal samples along the study duration. And again, we could find that the right foods were spiking at the right time when the people were consuming them, and all of these foods were represented by multiple unique proteins, making the signal very crisp and very trustworthy. Then reviewer number 2 kindly asked us to test the sensitivity of the method, so we had to do another clinical trial. We took a group of volunteers, we gave them elevating levels of peanuts. First 5 peanuts, then a washout, then 20 peanuts a day, then a washout, then 40 peanuts a day, and we could see a very nice dose-response curve. It's probably even sensitive below 5 peanuts a day, but we didn't measure. And then we went to disease states. For example, we went to children with recently diagnosed celiac disease. So when they came to their primary care physicians or to their pediatric GI doctors and diagnosed with celiac, they were all eating bread. So we performed our thing on them, and you can see the signal very high in all of these children. Then they were moved into a gluten-free diet, the first-line treatment of celiac, and the signal went down. Other than a single child that stayed up, so we went back to the child, and we found out that the child was cheating. So in general, this method is termed by us as IFOMED, Integrated Proteogenomics of Host Microbiome Diet. It enables to kind of quantify non-invasively and in parallel the 3 main players that impact many multifactorial disease, the host, the microbiome, and our diet. Just to show you, in IBD, which was the first multifactorial disease we went to, this enables to generate a very big separation, both of the microbiome and the host signals between diseased and healthy individuals in very modestly sized cohorts of around a few tens of individuals. Those of you who do IBD work like me and use the DNA shotgun metagenomic sequencing as our readout, we usually need hundreds and thousands of patients in order to generate an overlapping separation between diseased and healthy controls. So it means that function has a better meaning than composition in this case. We were able to uncover new ways or new forms of microbiome dysbiosis, of disturbances in the microbiome that are associated with disease, compositional dysbiosis in which whole signatures of proteins from a given commensal go down or up, so the abundance of that commensal is changing, or a newly found functional dysbiosis in which discrete cassettes of proteins are changed in a disease context in a commensal that doesn't change in its abundance. This is a facet that we were completely blind to until we directly measured function. And this, from a translational point of view, also enables us to identify new trans-kingdom biomarkers. For example, using our system on 2 previously published US-based IBD studies, also modestly sized, identifying dozens of candidate proteins that may be usable as biomarkers, and then comparing different random pairs of these candidate biomarkers against the gold standard and the only biomarker clinically approved in IBD, which is calprotectin, and applying them to 2 different cohorts in different countries, because this is how we try to be kosher and not to do overfitting. And the long story short, we identified dozens of pairs of biomarkers that outperform this calprotectin, and these are now spun out and tested in the clinic. But we're talking about nutrition today, so we looked at the nutritional signals. We found that there is around an 85% agreement between our measurable nutritional signals and the food frequency questionnaires. And just to end with one small example of how you can harness nutrition in a disease setup, we took a group of children with new onset Crohn's disease. And in children, the first-line therapy is often a supplementation with a diet that is composed only of amino acids. It's called elemental diet. So one would expect that our system, our mass spec-based dietary signal would be reduced because the mass spec cannot detect these individual amino acids. And indeed, this was the case. Following the initiation of this dietary supplementation, you can see a reduction in the signal. The slope of the reduction reflects the compliance of the children to the intervention. And what was cute was that in the same samples, from the same price, you can also see the inflammatory signals generated by the host and the microbiome. And there is a nice direct correlation between the level of compliance to the treatment and the reduction in the inflammatory signal in the same children. So to summarize, IFOMED, the Integrated Proteogenomics of Host Microbiome Diet, enables us to try and contribute towards a holistic and functional assessment of host microbiome dietary interactions in any multifactorial disease. From a translational standpoint, it may enable us to identify new biomarkers, to quantify diet and digestion. I've not gone into this for the sake of time, and identify therapeutic targets. And in getting back to where I started for, this measurement-based food quantification potentiates our ability to harness AI into precision medicine, and these are efforts that are undergoing as we speak. And so not to have a whole talk in a cancer conference without mentioning cancer, as you may imagine, we are very interested in applying IFOMED in trying to learn new insights into dietary and microbiome contributions to cancer. We're currently doing this as part of 2 exciting EU-based cohorts in colorectal cancer and hepatocellular carcinoma, and we're always ever happy to collaborate with more. So I'll finish by thanking the amazing members of my 2 labs, one at the Weizmann Institute of Science in Israel, and the other is at the National Cancer Research Center in Heidelberg, Germany, who have been engaging in these ambitious trials. All the many basic science and clinical collaborators, there are just too many to mention in one slide. Without them, we couldn't have done anything. The funding agencies that are making all of this work possible. And I think I'll stop here. Thanks.
Thank you a lot for this excellent presentation. So we have time for a few questions.
Amazing talk. So can sort of linking your earlier work that you talked about in the first talk with the able to profile diet in the stool, can you see the variation in human metabolism that you so nicely found 10 years ago play out in the peptides that you find in the stool? On the other hand, assuming you and I are different in our what the right food is protect us from glycemic index, does that play out differently if we eat exactly the same banana? Do I spike more than you or vice versa?
Yeah, it's a fantastic question. And this is exactly the effort we're trying to achieve as we speak. And the signals that are emerging are that this inter-individual variability and new aspects of it that we were completely blind to could be visualized using this very high resolution approach. However, it's a game of big numbers, and it's rather expensive. So we're doing it, and we are currently developing cheaper and more scalable methods that we hope would enable to quantify nutrition without having to go to the pain of performing shotgun and mass spec on each sample. So this is work in progress. I would also mention that we are increasingly interested in quantifying the big black box of this variability, which is digestion and absorption, which we're completely blind to in decades of work. And there are some cute ways.
Has to be part of the first part, right?
And so the variability there is quite remarkable. We were surprised to find it not only between individuals, but also between different nutrients within an individual. So this is another project that we're trying to follow up on.
And since I'm still standing here and I don't see anyone else over there, I'm going to ask another question. And that is, so this is not me, but my colleagues out there who are the microbiome skeptics, they always argue that microbiome changes are a biomarker for diet. To what extent is that true? In other words, can you predict the microbiome from the peptides you see? If I eat bananas, do I raise a specific microbe to oversimplify the whole thing?
Yeah, so I think, first of all, you're walking into an open door here. Obviously, I'm not a microbiome skeptic, but I'm one of the biggest proponents for the need to transfer from correlation, association, and prediction into causality. And so 99% of all the papers you read from our group are actually focused around the proof of causality and molecular mechanism. What I've presented today does not reflect causality yet, because the problem is so complex that decoding human nutrition microbiome interactions by transferring discrete food components into germ-free mice is just not feasible. So there we're using AI as a kind of a bypass in order to form predictions which then need to be validated in humans prospectively, and that is the form of causality that we can generate under these complex setup. But you're absolutely right. Once the data is generated, I think there are hundreds of different projects that could start on each discrete protein that we've discovered that would go towards causality.
Yeah.
Okay, so thank you again to both speakers of that third session.